Hello everyone and welcome back to Biology with Mrs. Evans. So in this video we are going to look at the process of DNA replication in a little bit more detail. So this first few slides are going to be a review of basically what we covered in the last video. So remember, DNA is made up of nucleotides, and because it's DNA, the sugar in DNA is deoxyribose. So remember, it is a phosphate that you have here is attached to the sugar, which is attached to the nitrogenous base. And this picture representation over here, this would be one nucleotide, this is another nucleotide, so on and so forth. Notice again that the nucleotides are drawn so that you know that adenine is going to pair with thymine. You can see how it would fit into basically thymine's structure. Cytosine, remember, is going to pair with guanine. Again, notice that cytosine is going to fit the structure of guanine. Remember, the base pair rule we know is because of Chargraph, and that states that adenine is always going to pair with thymine, cytosine is always going to pair with guanine, and we can use the base pair rule to figure out the percentages for each base based on a known percentage that we have for one of the four bases. We also talked about the DNA backbone in the last video. Remember, the backbone are basically the sides of our ladder, and the backbone consists of the phosphate and the sugar. And remember, they are going to be bonded together with a covalent bond. Remember, covalent bonds are our strong bonds, so you don't want your backbone basically to easily fall apart. So they are held together by covalent bonds. And then the rungs of the ladder, the inside, again right here, these are going to be held together by a hydrogen bond, and we want that to be a weak bond so that the cell doesn't have to spend too much energy to split those apart. And when it splits apart, you're going to have one side and another side, and each of those strands can be used to make copies of the DNA molecule. So at the end of the DNA replication, you're going to have one old strand and one new strand. And again, it's because of the base pair rule as to how easy it is basically for DNA to be replicated. So bonds, we already discussed. Hydrogen bonds between the base pairs, and again, that's because they're weak. You want it to be easily split apart. And then the bond here between the phosphate and the sugar is our covalent bond, and we want that bond to be strong so that when the two strands do split apart, it doesn't just basically shatter. It stays intact, and it can be easily copied through DNA replication. So DNA length. So we already talked about prokaryotes and eukaryotes and their shape of DNA, how eukaryotes have basically lots of DNA and it's in the shape of a double helix, and that we said that humans have basically 4 to 6 chromosomes in our somatic cells. And prokaryotes, remember, have a single circular strand of DNA. If you were to stretch out a molecule of DNA, it would be about 3 feet long. So 3 feet long, you should be able to see it, but we can't see it. So how is it that it can fit inside of a microscopic cell? And that is because, or it can do, it can basically fit inside a cell because it coils around a protein which we call histone. And I'll show you a picture here. So chromosomes are typically what they are tightly packed chromatin, and chromatin is simply basically threads of DNA that are basically DNA wrapped around a histone, and a histone is again the protein that DNA wraps around. So you see the strand of DNA and it's wrapping around these histones, and it basically coils really tight to form chromatin, and then that chromatin can even coil tight to form the chromosomes that we see during basically cell division. So here's a picture representation. This is something that you should know. These X-like structures are basically what we call chromosomes. It consists of a centromere, which is the center part here, and that centromere holds together two sister chromatids. All right, so this side would be one chromatid, and then this side would be another chromatid. And as you see, DNA is coiled around the histones here. And then what you need to know is that on chromosomes there are different genes, and they're usually basically you see them in these little like banding patterns, and those bands basically represent different genes that are on a chromosome. And we're going to talk about that a little bit more in detail when we get into cell division, which is Unit 8. So know that the protein that DNA coils around, these guys right here, is called histone. The thread-like structure is called chromatin, and basically, or you also see it, or also hear it referred to as chromatin, is the thread-like structure with DNA wrapped around a protein, which would be histone. So prokaryotes, again, prokaryotes have a single circular strand of DNA, and they also can have a smaller piece right here that's called a plasmid. And plasmids are basically smaller chunks of DNA that have been spliced together, and it's believed that these pieces of DNA is what allows for bacteria to become antibiotic resistant. So it's little chunks of let's say ancestral DNA that has passed down to future strands of bacteria that allowed them to become basically resistant to antibiotics. So what's on a chromosome? So here's another picture representation of what I was trying to just draw a second ago. These bands that you see would be the different genes that are located on that chromosome. And remember, a gene is basically a segment of DNA that codes for a specific trait. So let's say this particular gene right here would code for your eye color, and another gene, let's say this gene right here, codes that you are right-handed as opposed to left-handed. They're not actually on the same chromosome, but that's just an example of what that would be on a chromosome. So different genes are on a chromosome, and they all code for different things. So the code is based on the sequence of basically the nitrogenous bases. So this code would represent, let's say you're left-handed, and then there would be a different code for somebody that is right-handed. It's based on these nitrogenous bases, and we know that DNA is what makes you unique. No two people have the same DNA, with the exception of, remember there's always an exception to the rule, with identical twins. They do have the same DNA, but other than that, we're all unique, basically in the sequence of our nitrogenous bases as to our unique traits. So DNA replication. So we know that DNA is located in the nucleus. So that means that DNA replication takes place in the nucleus. So why does a cell need to copy its DNA? It needs to copy its DNA basically anytime it needs to make new cells. So right now, everybody in this class is growing. You guys are getting taller. I'm growing because my hair is growing. I'm not growing any taller, but I seem to be adding weight here lately, so I'm still growing. Repair. When you cut your skin or break a bone, all right, your body is going to need to repair that damage. In order to do that, it needs to make new cells. And before your body can make new cells, it has to replicate the DNA. So in order to repair DNA needs to be copied, and then also in reproduction, your cells are constantly basically making DNA if you're a guy in order to pass on some of your traits to your offspring. So reproduction, repair, and growth is when DNA needs to be replicated. So enzymes, there are several enzymes that are involved in DNA replication, and this is going to be a very simple overview of DNA replication. It is actually more complicated than I'm going to cover in this video. If you take this class in college or you take AP Biology, you'll see how complicated it is. For example, the DNA is basically the strands of DNA. One is going to be 5' to 3', and the other one is going to be the opposite. So this would be 3', and the other one would be 5'. Sorry, and the DNA replication is different depending on which strand is being copied. So for us, we're just going to make it a very simple process. So before DNA can be replicated, you know that DNA is in the shape of a double helix. So before it can copy, the DNA must unwind. So it needs to start to straighten out. After it unwinds, then the DNA, the double helix, needs to unzip. So that is when helicase is going to come in, and it's going to start to break apart those hydrogen bonds to expose the two strands. And then once the two strands are exposed, that's when DNA polymerase is going to basically come in and get to work. DNA polymerase bonds to little segments on DNA that are called promoters. That tells basically DNA polymerase where to bind and where basically it's going to start replication. So for eukaryotes, our DNA replication is, remember we have lots of DNA, so our replication can start in different places on our DNA strands. And basically what happens is where it starts, so this area right here, we would call this a replication fork, where basically the DNA started to split apart, and now you have DNA replication. And it can happen, you know, in 100 different spots on our DNA molecule, and that's because we have a lot of it to copy, so it speeds up the process of DNA replication. And DNA polymerase is going to bond to the DNA and going to read those exposed strands. So you see here, this is C. DNA polymerase would bring over a G and help bond that new base to the new, or to the old strand. And it's going to read a T, so it's going to bring over an A. It's going to read an A, and it's going to bring over a T. It's going to read a G, bring over a C. So DNA polymerase is going to read those exposed strands and then bring over the matching base based on what it reads. So again, it's a 4-step process for DNA replication. The first thing is for the DNA to unwind. All right, so if you ever had a phone cord get tangled up, it's very difficult. So in order to copy, it needs to unwind that DNA. Once it's unwound, then helicase is going to come in and start to break the hydrogen bonds to expose basically the two strands of DNA. Once those two strands are split, then DNA polymerase is going to come and bind to the strand and start to read it. It's going to make a copy. Remember, it's a complementary strands of DNA, so one strand of DNA can code for the other strand of DNA. And that's again because of the base pair rule. A bonds with T, C bonds with G. Once it's made a copy, then the hydrogen bonds are going to reform between the two, and at that point the DNA will start to recoil itself back into the shape of a double helix. So again, DNA replication, we refer to it as a semi-conservative, and that's because, remember, semi means half and conservative means to save. So the process is you have one old strand and one new strand for each molecule of DNA. So one old strand, one new strand, and that is again because one strand of DNA can code for another. So when this DNA splits, it's going to create two strands of DNA. Each one is going to have one old strand and one new strand. Alright, so DNA replication, you are going to be expected to tell the complementary strand for a segment of DNA, and we did this in the last video. So if I were to give you a segment of DNA that looks like this, and it could be anything, I could put GGCCAA, whatever, you would be expected to give the complementary strand. So again, the way you would do it is you're going to play the role of DNA polymerase. You know that A bonds with T, T bonds with A, C bonds with G, G bonds with C, C bonds with G, T bonds with A, G, T. Alright, so again, the strands are complementary, and this is a much more complicated process, but this is the gist of what you need to know for this level of biology. And in the readings that you guys did, you'll notice that DNA has, or chromosomes have, what we call telomeres on the tips, and those telomeres are to protect the important part of DNA. So they're basically nonsense DNA that's there, that if it gets cut off, it doesn't matter because it doesn't code for anything. And remember from the readings that you get in Unit 4, those telomeres are going to protect the DNA from being miscopied, and when the telomeres get too short, then DNA replication is going to stop because they don't want to take a risk of anything important being cut out. And the telomeres, we're learning now, have a lot to do with the aging process. So as they start to get shorter and shorter, we start to see basically people showing signs of basically their age, and that has a lot to do with telomeres, and more research is being done into those and the roles that they with aging. Alright, so we know from Unit 2 that DNA is only one type of nucleic acid. Another type of nucleic acid that we talked about in Unit 2 is RNA, and it's called RNA because the sugar in RNA is ribose. And again, you learned that in Unit 2. And the other thing that you learned in Unit 2 when you did the hands-on activity is that RNA does not have the nitrogenous base thymine. It is replaced with uracil. So that means that when RNA is made, and we already talked about this in Unit 4, when RNA is made from DNA, when adenine is read, it is going to bring over a uracil in order to make RNA, and we're going to go into that a little bit more in detail in the next unit. So, how is RNA different than DNA? Obviously, it has uracil instead of thymine. It has a different sugar. Remember, DNA has deoxyribose, and RNA is going to have ribose. And DNA, remember, is double-stranded. RNA is single-stranded. So when we talked about cell transport in Unit 3, we talked about basically things being able to pass through the cell membrane. And in Unit 4, we talked about the nucleus having a nuclear membrane. DNA is a double-stranded molecule, so it is too big to pass through the nuclear membrane, or remember, we also call that the nuclear envelope. So messenger RNA plays a vital role in getting the information that is stored in the nucleus to the ribosome, because remember, ribosomes make protein, and because they are single-stranded, they can pass through the nuclear envelope. So they're going to carry that message that they have basically copied from DNA, and they're going to take it to the ribosome, and the ribosome is going to read that message and then make the corresponding proteins that are associated with that message. So messenger RNA, there are different types of RNA. That's another difference between DNA and RNA. DNA is basically one type. RNA has 3 types. And messenger RNA is abbreviated with this little M that you see here. And again, it is single-stranded. It is going to carry the message from the nucleus to the ribosome. And the way that messenger RNA is basically made, it's similar to the process of DNA replication. DNA is going to split apart, as you see here, and instead of DNA polymerase reading the strand of DNA, it's going to be rRNA. So rRNA is going to read it, and it's going to bring over the matching bases. So again, RNA doesn't have T, so it read A, so now it needs to bring over a uracil to bind to the adenine. It read A again, so it brought on over another U, A again, so it brought over another U. This time it read C, so it brings over G. And once this strand of messenger RNA is made, it's going to leave the nucleus of the cell and travel to the ribosome, where it is going to be read and made into a protein. So messenger RNA is one type. Another type of RNA is what we call transfer RNA. It's usually abbreviated as you see there on the screen, TRNA. Transfer RNA, it is going to basically bring over an amino acid that corresponds to a segment of the messenger RNA. So if you know here, if you look here, you'll see that the CUA is on the messenger RNA. And what bonds with C? G bonds with C. What bonds with U? A bonds with U. So you'll notice that these match up to that segment of messenger RNA, and that corresponds to a specific amino acid that you see here. This would be leucine. So transfer RNA is going to bring over the amino acids to form the protein, and we're going to get into more detail about how that works in the next unit. And then the last type of RNA, this is a relatively new discovery as far as understanding how RNA works. The ribosome, remember, has its own RNA associated with it. So RNA is basically believed to play a role in protein synthesis because it's going to help basically the messenger RNA attach, if you will, to the ribosome, so that the ribosome can basically move down it and read the messenger RNA and tell basically the transfer RNA what amino acid to bring in order to form the protein that is being made. So what's important is the shape of DNA allows it to be easily copied. So remember, it has 2 strands, and the hydrogen bonds in the center allow it to be easily split, and the covalent bonds on the side or the backbone allow it to basically stay in one nice strand when they do split apart, so it doesn't end up with 1,000 pieces that on the side, because if that happened you wouldn't be able to copy it. So the shape allows it to be easily copied. Remember, it's called a semi-conservative process because each strand can be used to make a new copy, so that the new molecule of DNA has 1 strand of old DNA and 1 new strand of DNA. And you need to know the enzymes that are associated with DNA replication. Remember, helicase is going to split apart the double helix, and then DNA polymerase is going to read those exposed strands and bring over the matching bases. There are other enzymes that are associated with DNA replication. You'll get into that if you take this class in college or AP Bio, but those are the 2 ones that you need to know for DNA replication. You need to know replication takes place in the nucleus, because that's, again, remember, where DNA is stored in the cell. And you need to know the differences between DNA and RNA. DNA has the sugar deoxyribose. RNA has the sugar ribose. RNA is single-stranded, and it's single-stranded so that it can leave the nucleus and carry that message to the ribosome. DNA is double-stranded and cannot leave the nucleus. And DNA has the base thymine, and RNA has the base uracil. And then again, DNA, there's really only one type of DNA, and that's the shape of a double helix. And then RNA has 3 different types. You have messenger RNA, which is going to carry the message from the nucleus to the ribosome. You have transfer RNA, which is going to bring over amino acids to the ribosome based on the messenger RNA strand that is being read. And then ribosomal RNA, we now know, plays a role in basically messenger RNA attaching to the ribosome. More detail will probably come out over the next couple of years as more is learned about ribosomal RNA, but that's basically nucleic acids in a nutshell. So those are the basics about DNA. Again, this is a much more complicated process than what was covered in this video, but these are what you need to know for this level of biology. If you take this class in college, or if you take AP Bio, you'll get into more specifics like telomerase is used to make sure that those telomeres are still on the chromosome to protect the chromosome as it's being copied, etc. You'll learn how you have the leading strand and the lagging strand and all of those things associated with that. So these are the basics. Hope you have a great day, and I'll see you in class. Bye.